I will now start with giving the floor to the first speaker, which is Marco Cavalieri. So up to you, Marco.
Thank you so much, Noel, and good afternoon to all of you. So in my presentation, I will give you a little bit of an overview of what happens during the development phase of vaccines. And I will also take the opportunity to mention what is unique for COVID-19 vaccines versus what, on the other hand, would be exactly the same for COVID-19 vaccine evaluation as for any other vaccine that we have been authorizing in the past. So my focus indeed will be on the left side of this picture, and I will talk to you about these three domains that are really the pillars of vaccine development, starting from the pharmaceutical quality and then moving into the non-clinical testing that occur for vaccines under development, and then the very important areas of clinical trials, while colleagues, as Noel said, will talk to you later about the evaluation process and the post-authorization activities. As you know, vaccines are medicinal products that essentially trigger the immune system, so our body's defense against external organisms, so that once we encounter the specific pathogens, we can be protected and defended against it. The first step at the beginning of the process is identifying your vaccine construct. So what happens is that, first of all, the technology that will be employed has to be selected in order to make sure that you will have this immune response that could be protective. And also it has to be decided which component of the virus, so in this case, the SARS-CoV-2 virus, will have to be incorporated in the vaccine in order to elicit an immune response that could be effective. All these studies are conducted in the laboratory at the beginning of the development. And throughout all the development phase, there will be more and more activity related to pharmaceutical development that will include a good characterization of all the components inside the vaccine and their purity. Also, in vitro tests and assays will be set up in order to determine rapidly what is the biological activity that the vaccine is exerting. And also, starting from the small scale production that occurs in the early phases of research and development, then gradually move up to larger amount of vaccines produced in each single batch, and with the need to incorporate good understanding on how it can be controlled at each step of the manufacturing phase, and to have a test that can control that what is manufactured is exactly what we expect to see, and to ensure at the end of the day that each of the batch of the vaccine is manufactured in a consistent way and is of good quality. Also importantly would be to start as soon as possible with studies that will look into the stability of the vaccine at different temperatures, as this, as you know, can be extremely important and even critical at times in order to ensure that adequate vaccination campaign can be carried on. The second important domain that has to be considered is around the so-called non-clinical development. So this is a set of laboratory tests that are done before the vaccines is tested in humans in clinical trials. And essentially what is looked at is, first of all, what type of immune response is elicited by the vaccine in animals. And importantly, to try to understand whether the vaccine may cause any safety problem when administered to animals. So this is in the context of the so-called toxicity studies. But also there are some specific toxicity studies that are conducted in order to be sure that the vaccine does not cause fertility problems or is not affecting the baby's development before birth. And these studies are important before the vaccine can be used in pregnant women or childbearing potential. Another important area is conducting studies that are called challenge studies, in which is it checked if animals, after they receive the COVID-19 vaccines, are protected from the disease when exposed to the virus deliberately, and also importantly, to make sure that the vaccination is not causing any paradoxical effect of worsening the disease, as this is a signal that was seen in preclinical models with some investigational vaccines for other coronaviruses, such as SARS and MERS. Also, for some vaccine, it is also very important to do some preclinical study that would determine how the vaccine is spreading around the body. So whether it's not just staying around the injection site and the lymph node in the surroundings, but also maybe spread to other parts and other organs. And this could be quite important also for toxicity studies and to have a good understanding of the behavior of the vaccine when administered in humans. Then the next important phase is the clinical development. Clinical trials are studies done in human which would show how safe the vaccine is, so looking into the safety of the vaccine, how well the vaccine works, so the efficacy of the vaccine, but also to determine what is the immune response that is elicited by the vaccine in human beings, the so-called immunogenicity. And these studies are divided in 3 phases, starting from phase 1, which are the early studies and the first into human clinical trials, and then moving up into phase 2 and the large phase 3 trials that will be potentially pivotal for approval with a much larger population involved. All these studies should follow strict scientific and ethical rules and have to be approved by the competent authorities in charge of the clinical trial approval and by the ethics committee in order to safeguard the safety and the rights of the participants in the trials. So going a bit into more details, the phase 1 studies are really the early trials and the first into man trials. Normally we're talking about a limited amount of healthy volunteers included, and what is measured there is what is the immune response to the vaccine, whether the vaccine is safe and can be moved into larger studies with more volunteers included, and importantly, what are the doses that really are showing a good immune response and an acceptable safety profile, and therefore can be selected for larger clinical trials. The phase 2 studies normally involve several hundred volunteers, and they would confirm what would be the best dose to be used, looking in particular at the most common side effects that would occur in such studies, and also having confirmation of the immune response in a large data set and larger than what we've seen in phase 1 trials. And then we move to the phase 3 trials, which are the pivotal trials that could support authorization. Here, normally these studies are designed involving thousands of volunteers. And essentially what is measured is how the vaccine protects against the disease compared to placebo, which is a dummy, or in few cases also with a non-COVID vaccine that could be used as a comparator. Also, these studies are very robust in order to determine the safety of the vaccine and also to determine what is the occurrence of less common side effects. Moving into COVID-19 vaccines specifically, the EMA has been engaging with the developers and has given scientific advice around what would be our requirement. And also recently, we published a reflection paper that summarizes our thinking around what would be required in these clinical trials. First of all, we would expect that the primary endpoint of these clinical trials would be prevention of symptomatic disease as a reliable and appropriate measure of benefit of these vaccines. This would mean that the vaccine should show that it's causing less disease with symptoms in people that were vaccinated compared to those that received placebo. Other secondary endpoint will collect evidence around important outcomes such as severe disease and may try to determine the efficacy in terms of protection from infection. However, it is clear that despite these studies are quite large and quite comprehensive, they will not be able to answer all the questions that you may have. In particular, what these studies might not be able to do at this stage is to give us sufficient information about the long-term protection that they can generate. And as said, they might not be able to provide good evidence around the level of protection from infection, so protecting from asymptomatic infection, and also will not be necessarily informative with respect to the potential of the vaccine to block transmission of the virus in the community, which is, of course, a very important endpoint from a public health perspective, and this will determine how to use the vaccine and what kind of coverage will be required in order really to stop the transmission of the virus and put an end to the pandemic. For all of these very important questions, there is a need of conducting studies in the post-approval phase, and that's what we are already discussing with other players from a European perspective, and also with the companies, as it will be expected that they will have to conduct studies in this regard. The EMA in the reflection paper made clear that in terms of the expected efficacy, we cannot really say upfront what is the minimal level that would support a positive benefit risk and a potential approval, as indeed it will all be a matter of real evaluation, the benefits against the risk, and really understanding whether this balance is positive. And therefore, we were not able to put up front precisely any specific number. However, we agreed that these studies have to be powered in order to generate robust evidence of efficacy and safety, and therefore we agreed with sponsor that these studies have to be designed in order to show an efficacy of 50% or more. 50% efficacy means that the vaccine would prevent half of the cases of symptomatic COVID-19 compared to placebo. Now, you have seen already that some of the vaccines have already declared success in their clinical trials and issued press releases that will show the efficacy to be 90% or more, which would mean that 9 out of 10 cases of symptomatic COVID-19 would be prevented by the vaccine compared to the placebo. As said, these clinical trials are quite large, often in the order of 30,000 subjects included in such trials, or even larger. When discussing with developers, we made clear that we would like to see ideally 1/4 of all participants above 65 years of age, as we know that age is a very important risk factor for mortality and morbidity due to COVID-19. Also, the inclusion of people with underlying conditions such as cardiovascular disease or diabetes has been strongly recommended, as we know that the risk of severe disease also in this population is higher. Some of the studies also include adolescents above the age of 16 years. However, for younger children, we expect that the studies will be conducted only once we have a good picture around the efficacy and safety of the vaccine in the adult population. But we would expect, and has been agreed in the pediatric investigational plan, that such development will have to take place. Ethnic minorities have been included in these trials, and we, of course, agreed on the inclusion of such groups. Of course, it will be very difficult to be comprehensive, but at least we have seen really genuine efforts to include such minorities in the studies. From a safety perspective, the follow-up is expected to be at least of 6 weeks long after the last dose of vaccine has been received. And this is because we know that most side effects would occur within 4 to 6 weeks after having received a dose of vaccine. However, these trials will last for 1 year or even longer in some cases, which potentially will give us invaluable information on the long-term protection that could be provided by the vaccines, and also whether some side effects might occur later on after vaccination. Of course, the enhancement of disease being one of the important areas that we would like to collect data. Whether participants in clinical trials can be kept onto placebo long-term after a vaccine is authorized and made available in any specific country or location, that is something that, of course, we cannot predict, and we might expect that it might be challenging and even not ethically feasible. However, we are urging companies to try their best in order to make sure that these studies are continued in a rigorous way so that we have good evidence in the long run. An important point that has often been raised is that COVID-19 vaccines seem to be going too fast. Now, what you see here on this slide is what normally would happen for a vaccine under development in non-pandemic scenarios. What you see that a lot of the activities that I described to you before related to the various phases of development are occurring in sequence, one after the other, and often with intervals and pauses between each phase. And this has led to an average of 5 to 10 years before a vaccine can be ready for approval, and then once approved, being deployed after manufacturing on a large scale. What we are seeing here with COVID-19 is that all these steps are still done and to a rigorous way, but everything is much more compressed, with a lot of these activities occurring in parallel or with respect to the clinical phases according to a seamless approach in which there is a continuum between the phase 1, the phase 2, and the phase 3 trial. This has allowed something that is being unprecedented, which is seeing a vaccine developed in a matter of months instead of years. However, it's important for me to stress that all this is happening without cutting any corner in the data generation. At the end of the day, we will have the same amount of evidence that we would have for any other vaccine developed in standard conditions. And indeed, also from our side, the regulatory standards are not changed. So the same standard in terms of safety, quality, and efficacy that would normally apply to any other vaccine will be applicable here for COVID-19. And as said, here what we're seeing is just really a compression in time of all the activities with respect to what normally happens, but really no decrease in the amount of evidence generated. This has been possible because there has been an incredible effort by all the players and the stakeholders to mobilize resources simultaneously and make sure by a number of agreements and collaboration to make sure that this vaccine could advance very rapidly, and also that they have sufficient fund to make sure that all these activities are carried on in really a very short time frame. From the EMA perspective, we made also a contribution in this sense and really foster how rapidly this vaccine could have been advanced. And this has been done essentially by establishing the EMA COVID-19 task force according to our ELFREC plan, which is a group composed by EMA staff and experts from the regulatory network in the area of vaccines and infectious diseases, and with other expertise as well, that has helped in having a continuous and rapid dialogue with the developer so that we could provide feedback in a way that normally is not done in a continuous fashion, and also giving rapid scientific advice that has been instrumental in directing developers very rapidly on what would be the requirement from a regulatory point of view. The other very important peculiarity that has happened here with COVID-19 is that manufacturers and sponsors have decided to start scaling up the manufacturing much before they were aware of whether their vaccine is successful in large clinical trial. So in other words, starting scaling up at risk. This can be extremely important in making sure that once the vaccines are approved, there will be sufficient amount to be deployed in order to have an impact on the pandemic in the fastest possible way. The last point I want to cover here is which vaccines for COVID-19 are currently under assessment for approval by the EMA. And I have to say that we got 4 vaccines that officially started rolling review and 2 also submitted officially for conditional marketing authorization. 2 are messenger RNA vaccine that contain genetic instruction in the form of messenger RNA for making an immune response against the SARS-CoV-2. One is from Pfizer-BioNTech and the other one from Moderna. Then we have two viral vector vaccine, which use this modified and harmless adenovirus, human adenovirus or chimpanzee adenovirus. And these viruses, they carry the genetic instruction for making an immune response against the SARS-CoV-2 as well. This vaccine could advance very rapidly for a number of reasons. Of course, this technology might be more prone for rapidly advancing them in the early phases of development. And they are based on the experience that was already with these companies with the same platform technology, but with other pathogens and with vaccines that were already under development. However, these are not the only vaccines with whom we are talking. There are many other vaccines that came for discussion to the EMA, and a few more could be in the position of starting very soon a rolling review with the EMA for a potential approval in the near future. So if I can conclude, with COVID-19 vaccine, we have the same types of studies as for any other vaccine. The only difference is that the timelines for the development have been shortened, and this thanks to the pooling of expertise, resources, and funds that made this possible. The studies that will be pivotal in supporting a positive benefit risk are large clinical trials with a rigorous design and would generate robust evidence for our deliberations. The expected benefit that we will see at time of approval will be demonstrated in the reduction in COVID-19 disease mainly, and we have to accept that there will be uncertainties at that point in time, particularly with respect to the long-term protection and what could be the ability of the vaccine to turn down the community transmission. Also, important evidence on safety will be available that would allow a robust conclusion also in that sense. Let me also say that the use of face mask, hand hygiene, and physical distance would remain important public health measure, at the very least until large amount of vaccines will be deployed and the vast majority of the population would have been vaccinated. And even then, we still don't know exactly what will be the impact. So it's very important that we maintain a rigorous approach in order to protect public health. And the last important statement from my side is that the high regulatory standards for quality, safety, and efficacy are here maintained, and that comprehensive data packages will be submitted in order to support the conclusions of the EMA. So this is the end of my presentation, and now I will hand over to Dr. Fergus Sweeney, who will talk to you about the regulatory process for evaluation and approval of vaccines at the EMA. Thank you.